So I want to speak about imperfect plants in an imperfect world. And I hope you all wonder what I mean with this. And I'm referring to a saying in English, goes back to Thomas Weisern, who said, "One man's poison, another's meat." Put differently by an important American philosopher much more recently, Yogi Berra, was if the world were perfect, it wouldn't be. Now, you might think I'm a bit of a joker here. What do I really mean with this? I became interested in studying evolutionary biology 25 years ago or so for a very simple reason, that in evolutionary biology, it's all around trade-off, and that something that is beneficial in one setting often is not beneficial in another setting. And that's why it's impossible to reach perfection. And some of this evidence actually comes from human genetics. When we think about human disease, we typically think about mutations that cause disease, that make us unhealthy. But there's a lot of evidence that many of such mutations actually also have positive attributes. So you might have seen, for example, there's a connection between Huntington disease and having a lot of offspring. And some of the clusters where they actually first investigated Huntington's disease and were able to map it, they come actually from founders who had well over 10 children. So because things really depend on context a lot, evolutionary biologists have coined all kinds of different terms for phenomena that are context dependent. One of them is epistasis, where different genetic backgrounds will modify the impact of a mutation. And that's much of what I'm going to talk about today. There is balancing selection, where it matters what neighbors you have. So for example, when it is about defending against pathogens, it often pays to be different from everybody else, because then there's not much incentive for the pathogen to go after you because you are really rare and it's difficult to make a living on that particularly genotype. Antagonistic selection, which means that in different environments, the same allele might have either a positive or a negative effect. So an allele that is positive when it's hot might actually be detrimental when it's cold. Antagonistic pleiotropy, where the same gene has either positive or negative effects depending on the trait. A good example in plants is flowering and water use efficiency, where alleles that accelerate flowering often also reduce water use efficiency. And then finally, at the most complex level, eco-evolutionary feedbacks, where actually an allele eventually leads to changes in the environment. That's a different ecosystem structure. And so an allele that initially was a good allele because it changed the environment does not become so advantageous anymore. Now, in many organisms, including humans, one aspect of fitness is typically body size. And you might think, well, obviously in humans, being big is better, as exemplified by this fine specimen. But again, it really depends on context. So see this jockey here, obviously in his profession, it's much better to be of slight stature. And so quite a few years ago, almost 20 years ago, Marco Tedesco, who is now an assistant professor at UBC, he became fascinated with the simple fact that when you grew different accessions of Arabidopsis thaliana in the lab, that their size varied quite a bit. And let me see, can you see my cursor?
Yeah.
Yeah, okay. So, in particular, there was one group of accessions characterized by the accession EST-1, which comes from Estonia, where plants were small, made leaves more slowly, and then the leaves often suffered from this late onset necrosis. He and Suresh Balasubramanian, who is now a professor at Monash, proceeded to map these genes by different means, including what was the first successful GWAS in plants, and they found that there was a single locus underlying much of the variation in size and these other traits, such as slower leaf production and so on. And this, of course, was enabled by the 1001 Genomes Project. And this single hit was at a gene called ACD6. And indeed, when you look at various accessions that have this particular allele, ACD6 EST-1, when you knock down this allele, you can see that in all these cases, the plants become bigger and often much, much bigger. And when you look at the cell death that is associated with the small size, you see rampant cell death from staining dead cells with toluidine blue in the wild types, but in all the lines where the ACD6 EST-1 allele was knocked down, the cell death disappears. Now, this seems somewhat odd that quite a good fraction of Arabidopsis accessions, 10 to 20% in wild populations, have this allele, which makes them much smaller. But obviously, necrosis was a giveaway. You are a plant biologist, and I'm sure you all know that necrosis is actually a typical immune response. So it wasn't very difficult to come up with this hypothesis that perhaps the specific allele here, ACD6 EST-1, provides protection against pathogens. And indeed, that turned out to be true. So this is just one particular pathogen, a naturally occurring pathogen, Arabidopsis thaliana, a fungus, Scolovinomyces aurantii. And the EST-1 strain, where we first found this allele, is completely immune to it. The reference strain, Columbia 0, you can see can be infected quite well by this fungus. And then when we knock down the ACD6 gene in two different lines, you see that they now also become susceptible to infection by this fungus. Now, infection, by one specific pathogen strain, that's not, that impressive. It did become more meaningful when we tested other pathogens, including an oomycete, bacteria, even insects. And in all cases, the ACD6 EST-1 allele conferred greater resistance. So this seems to be a typical growth defense trade-off where these plants are smaller, but at the same time, they are better defended against a wide variety of pathogens. Now, we got into necrosis at around the same time in a very different way as well. So here I first discussed an example of necrosis that we find in inbred strains that is conditioned by alleles at a single locus, ACD6. So at around the same time, Janne Lempe, and then soon followed by Kirsten Bomblies, who's now a professor at ETH, they found these plants that were F1 offspring of otherwise normal-looking accessions of Arabidopsis thaliana, or was seen here on the top or on the bottom. And these F1 offspring were much smaller, and they also exhibited sign of necrosis. And so initially, we didn't know what was going on. We looked at gene expression back then that was still with microarrays. And even though the individual F1s here had quite different phenotypes, not that well visible at this resolution, but you already see that in size they differ quite a bit. These gene expression programs were quite similar. And the only genes that were enriched, particularly among the genes that were induced, were genes that were related to immunity. So broad induction of immune response. And since these plants hadn't been challenged by pathogens and this phenotype also appeared when we grew plants sterilely, this indicated that, again, we were dealing here with autoimmunity. So similar to this ACD6 EST case, but now in F1s, and often much, much more severe than we will see in these natural accessions. And so this phenomenon, necrosis that appears in hybrid or hybrid necrosis, is actually quite widespread. There are hundreds of cases described by breeders and naturalists in both crops and wild species, within and between species. And often, that will become important later, has often simple genetic architecture. But quite remarkably, even though this phenomenon had first been described in 1924 already, so 100 years ago, we were really the first ones to make this connection between hybrid necrosis and immunity. And it's quite a nice tale here about blinders that scientists sometimes have. There was just this notion until a long time ago that basically immunity you would only see after infection. And so this idea that you could have activation of immunity by purely genetic means was not well received until around the turn of the millennium. A few years later, On-Young Choi, who is now a professor in Oxford, joined the lab, and she said, interesting, these cases of hybrid necrosis here, but is this something that we can study systematically? How many cases are there actually in the Arabidopsis population? So we became Arabidopsis breeders. We had just sequenced the first 80 strains of Arabidopsis thaliana accessions from 8 different geographic regions, and then under leadership of On-Young, team in the lab proceeded to make crosses between all these strains, so over 6,000 crosses, over 3,000 unique combinations, and you can see that hybrid necrosis is quite widespread. The blue cases are the ones that are the mildest, and the purple cases or the magenta cases here are the most severe ones. And the only thing that is really important here, A, that it's widespread, and B, that you see it both within regions and between regions. Because at that time, we had this whole genome information. This is why we used these 80 strains. It was relatively straightforward to identify the causal genes for all the more common and more severe cases. So in total, 27 cases that we looked at. And so what we found here, these are the 5 chromosomes of Arabidopsis thaliana, that in many cases, the causal genes map to regions of the genome that were enriched for these NLR genes, nucleotide binding site loose enriched repeat receptors, a particular class of receptors. And I just remind you in the next slide what these are really about. But it makes sense. We see immunity, then often the causal genes map to genes that are known to be involved in immunity. And you have all kinds of genetic architecture. So here's a case that we like for a really obvious reason, where at a single locus, DM2, there were 5 different risk alleles, and each of these risk alleles interacted with a different allele somewhere else in the genome. Here we have alleles at the same locus, either at RPP45 or ACD6 again, that interact with each other. And this is my favorite case here, where we have 3 alleles at this locus that interact in an allele-specific manner with 3 alleles at this locus here. And I'll tell you more about this one. So again, this all made sense. On the one hand, enrichment for these NLR genes, and then we also already knew that NLRs are the most diverse genes in plant genomes, and some of this information came from an earlier paper that we had published in 2007, where we have used these massive microarrays with 1 billion, 10 to the 9 different oligonucleotides to interrogate the Arabidopsis thaliana genomes in 20 different accessions. And the NLR genes really stood out because they were so much more variable than any other family of in the genomes. So what do these NLR immune receptors do? Just a little reminder how immunity works in plants. You have pathogens, and pathogens in the first line of defense are being recognized through pathogen-associated molecular patterns, or PAMs, by PAM recognition receptors, or PRRs. And upon recognition of these PAMs, then immunity is induced to a limited extent, but enough to limit the proliferation of the bugs here. To that circumvent recognition, bacteria, but also fungi or mycete, also animals, they inject into plant cells their own proteins called pathogen effectors, which typically subvert recognition. Immunity is reduced again, and then the pathogen can proliferate again. We go from pathogen-triggered immunity to effector-triggered susceptibility. And this is where the second line of defense comes in, these NLR immune receptors, which recognize pathogen effectors, and they then activate immunity in overdrive, if you will, and limit, again, the pathogen. The next cycle, we go to effector-triggered immunity. The next cycle is that the pathogen will actually get rid of the recognized pathogen effectors, will deploy a different set of pathogen effectors, and again, you get pathogen proliferation, and then new NLRs evolve, and so on and so forth. And this general idea was popularized in a review published by Jonathan Jones and Jeff Dangle, together with Brian Staskowitz, recent recipients of the Wolf Prize for agriculture. So what is behind this hybrid necrosis? So in one case, DM1 and DM2, both of the causal genes encode immune receptor. Fran, who is now at the Institut Gustave Roussy, Paris, she showed that DM1 proteins form oligomers, DM2B form oligomers on their own, but when they come together, they can form these heteromeric oligomers. Now, these oligomers do what they are supposed to do, and that is, in the absence of pathogens, they don't do anything, so signaling is off. But these heteromers, even when there's no pathogen around, somehow the interaction is not quite proper, and they are in the on state, and therefore they signal the presence of pathogens when there are no pathogens around, and you get necrosis. I already mentioned this other case where we have allele-specific interaction between RPP7 and RPW8, and the genetic side of this was figured out by Christina Barragan, and then Lei Li, who has his own lab at the Chinese Agriculture, at the Chinese Academy of Science, together with Annette Habring in the lab, showed in this case that one of the causal proteins is RPW8 HR4, a protein from phase 0, interacted with an NLR immune receptor RPP7B to induce formation of a higher order complex. So in this case, the NLR protein does not form a complex without the ligand. And so if you have an HR4 homolog that doesn't cause necrosis, you don't get this higher order oligomer complex here. So how do we interpret this? Well, these HR4, these RPW8 HR4 proteins, we know that they make holes in the membrane, make these pores, and thereby kill cells. And they are apparently deployed by the plants in defense against pathogen. The plant cell kills itself basically by expressing these proteins. And we hypothesize that the function of these HR4 proteins can be inhibited by a yet unknown pathogen effector that somehow modifies the HR4 proteins. And that's in line with what many people have shown for how NLR proteins and the effectors that are being indirectly recognized by NLR proteins work. And then the RPP7 NLR protein would recognize this modified HR4 protein and then signal. What happens in the case of HR4 phase 0 is that even when there is no pathogen around, for some reason, this HR4 phase 0 version of the protein looks to the RPP7B receptor as if it had been modified by a pathogen, even though this is just a genetic mutation. We know that it's structural differences that are responsible for differential recognition of HR4 phase 0 versus canonical HR4. So what has that told us? Well, when you think about what happens in a field, you have many, many different pathogens, and an individual plant will be resistant to a few of these pathogens. But then when you look at other plants, they are resistant to a different spectrum of pathogens. So if we look at the entire field, we see that while individual plants can only resist some of the pathogens, pretty much all of the pathogens can be resisted at least by some of these plants. So if in principle, it's possible to recognize every pathogen, then why don't we have these super plants here which recognize every pathogen? And we think the explanation is you get autoimmunity because there is a limit to how many NLR immune receptors you can encode in your genome, because the more you encode, the chance goes up exponentially that there are either inappropriate interaction between NLR proteins themselves or between NLR proteins and other proteins in the plant cell, and these then cause autoimmunity. So there is a balance between how much immunity you can potentially have and avoiding the risk of autoimmunity. So I told you about necrosis. I told you about hybrid necrosis. The question is there also evidence for something like anti-hybrid necrosis, where you can suppress necrosis through interaction with proteins encoded elsewhere in the genome? And sorry, going forward. And indeed, there is. Chris Seidem, who now works for TradeSeq in Cambridge, she had been looking at accessions that have this ACD6 EST allele. And what she noticed was that, yes, many of them had necrosis, but there were also quite a few lines that had this allele, and they didn't have necrosis. And when they then just cloned out the ACD6 EST allele from some of these accessions, such as PRO0 or ARMXA180, and then transferred the ACD6 EST allele into a different background, she nicely saw she saw the small stature and necrosis. So the ACD6 EST gene that is present in these accessions, such as PRO0 and ARMXA180, is perfectly able to induce necrosis and small stature, but there must be other factors in the genomes of these accessions that prevent this. And then Wang Xingzhu, who now has his own lab at the Chinese Agricultural University, used GWAS to identify these, what we call modifiers of hyperactive ACD6 or MHA genes. And this is really the GWAS that I'm most proud of, where we used the 1001 genomes resource to identify accessions that have the ACD6 EST allele, phenotype these 84 accessions, and then ran a GWAS on this. And you see very, very nice hits to LOSA MHA1 and MH2, and today I'll only talk about MHA1. And it turns out that MHA1 encodes a small protein, and then also has a paralog, what we call MHA1-like or MHA1L. These are MHA1 homolog from other Brassicaceae. This is MHA1 in Colombia, which is the same as an EST-1. And then in one of these suppressor strains, Ti0, you can see Ti0, there's a single amino acid change and in effect residue that's otherwise conserved in ACD6 alleles and also in other species. And then MHA1L, there are quite a few differences. Notably, this position is also different in MHA1L and actually has an amino acid that's quite similar to the one in Ti0, threonine and a serine here. Further, an investigation showed that MHA1L is a primary mediator of pathogen responses, so not MHA1, but MHA1L. So MHA1L is induced when you infect plants with Pseudomonas DC3000. When Wang Cheng knocks out MHA1L, there is a reduced response to PAM, flag 22. There is an increased response as measured by reactive oxygen species when you overexpress MHA1L. And this is also reflected in pathogen resistance, where either a disarmed pathogen or fully functional pathogen proliferates more in MHA1L mutants and proliferates less in MHA1L overexpressors. And then finally, when you overexpress MHA1L, especially at 16 degrees, you get these very small and necrotic plants. So somewhat similar to what we saw in these ACD6 EST plants, for example. The interaction between ACD6 and MHA1L and MHA1 is allele specific, as you might expect. This is a GST pull down. And what this shows is association between the ankyrin repeats of ACD6. I hadn't mentioned this before, but ACD6 has transmembrane domains and N-terminal ankyrin repeats, and these ankyrin repeats can dimerize. And when you compare this one here, you see that MHA1L increases the dimerization of ankyrin repeats from Colombia version of ACD6, but not from EST version of ACD6, and that goes hand in hand with MHA1L genetically interacting with the Colombia version. So if you overexpress it in the Colombia background, you get these small necrotic plants, as I also showed in the previous slide. But when you overexpress it in the EST-1 background, nothing happens. And similarly, also also allele-specific interaction of MHA1 with ACD6. So this was this initial suppressor that we had found in the Ty0 strain. And again, we are looking at dimerization through the ankyrin ring. And you can see here when we have MHA1 EST-1 and looking at the EST-1 version of ACD6, and you see there is decreased dimerization when the Ty0 version of MHA1 is used versus the EST-1 version, whereas here with the Columbia versions of the ankyrin repeats of ACD6, it doesn't make a difference whether you add either EST-1 MHA1 or Ty0 MHA1. So this one does not react to MHA1, whereas ACD6 EST reacts strongly to MHA1. So summarize this in a little bit simplified slide here. So at ACD6, we have two different versions, Columbia and EST-1. This one reacts strongly to MHA1, whereas EST-1 does not react to MHA1 at all. And then MHA1L is this paralog, which comes in two flavors, either the Columbia version, which is the same as the EST version, and then the Ty0 version. And the only interaction is that Ty0 suppresses the activity of the EST-1 version of ACD6. And we'll get to what this really means at the molecular level in a few slides. So ACD6 is really the primary downstream effector of MHA1L. Wang Shen carried out a suppressor screen exploiting the fact that 35S MHA1L on its own causes these small necrotic plants. And then when he has these suppressors, the phenotype goes away. And this is just shown when you have a canonical ACD6 loss of function mutation, totally suppressed phenotype. If you have an ACD6 gain of function phenotype, is actually even further enhanced. What is important here is that many of the suppressor mutations that Wang Shen recovered are missense mutations. And many of the missense mutations are the ACD6, are in the ankyrin repeats. And they affect residues that are actually shared with these three proteins. What are these proteins? Well, these are proteins that are ligand-gated ion channels in animals or fungi. And there's just a sequence alignment between ACD6 and these three ion channel proteins. And this is just a superimposition of modeled ACD6 structure and the known TRPA1 structures. The ankyrin repeats are quite similar. The membrane portion is different. So we know that ACD6 interacts with MHA1 and MHA1L, which are small peptides. It is similar in structure to known ion channels. And when you mutate residues that are conserved in known ion channels, you lose ACD6 activity. So not too difficult to come up with this hypothesis that ACD6 is perhaps a peptide ligand-gated ion channel. And indeed, this is a collaboration with the Ludovic lab. First work in xenopus oocytes showed that you get currents when you express ACD6, and the currents are higher. Calcium-induced currents are higher with the gain of function mutation ACD6-1. And when you add MHA1L, you can also enhance the activity of ACD6 in this assay, but not when you have a loss of function mutation. Now we collaborated with the He and Pei labs at Duke, and they used a different orthologous system, also again animal cells, HEC293, with the FRUA2 calcium indicator. And again, you see that when you express ACD6, you get calcium influx. When you have the gain of function mutation, you get even more calcium influx. And again, when you add MHA1L, this activity of ACD6 is enhanced. And then the final piece of data, this is again from Wang Shen's lab at Chinese Agricultural University, work done by his postdoc Junbin Chen, who showed that the phenotype of these ACD6 gain of function mutants is calcium dependent. If you grow the plants in a calcium-limited environment, the phenotype is largely suppressed. And this is just a positive control a mutation in the different calcium channels. And then when you look at aquarin, calcium indicator luminescence, you see that it behaves as expected, that you have when you inactivate MHA1L, for example, here, or knock out ACD6, less calcium influx as indicated by this reporter, and more if you have a gain of function mutation or express MHA1L. And this is just the same data shown in a different way. So to summarize this, ACD6 apparently indicates a calcium channel. In the Columbia version, we have the protein sitting in the membrane, not oligomerized in the resting state, no calcium channel activity. When pathogen is being recognized, for example, FLAG22 through the BAC1 FLS2 heterodimeric receptor complex, MHA1L expression is induced, and then the small peptide leads to oligomerization of ACD6 through the ankyrin repeats and calcium influx, and you get immunity. You can achieve a similar effect by just overexpressing MHA1L from the 35S promoter. You get, again, dimerization of the protein through the ankyrin repeat of ACD6, calcium influx, and now autoimmunity because no pathogen is present. The same thing can also happen spontaneously when you have the ACD6 EST-1 version, which spontaneously oligomerizes, and therefore you get calcium influx. And that in turn can actually be prevented in a background where you have this T0 version of MHA1, which also interacts with the ankyrin repeats, but prevents the oligomerization. So that's the opposite of here, the pyrologous ligand, which favors oligomerization. Here it's a suppression of oligomerization. We don't know what MHA1 does otherwise, the other versions of MHA1, what they do outside of this peculiar interaction with EST-1 version of ACD6. All right, so I showed you this, what I think is really quite beautiful story of interaction between different proteins that lead to autoimmunity. There's natural variation. There's also natural variation in these interacting proteins. So this all suggests that this is important in nature. But to show this, we really have to go to nature. So a number of years ago, we said, let's go out there and start from the basics. And this is just ask, what kind of microbes do we actually have in and on Arabidopsis plants and wild Arabidopsis plants? And this was work led by Rebecca Schwab, who is a group leader in my department here at Max Planck, and Talia Karasov, who is now a professor at the University of Utah. So we went all over Europe and into Asia and sampled Arabidopsis plants in a single year from the Mediterranean to Scandinavia and from the Atlantic coast to the Caucasus. And we were quite surprised looking at the microbiome in the leaves of plants and the above ground portion was that they nicely clustered into two different groups, a southern cluster and a northern cluster. The individual families that we have in each of these clusters are not so different. The differences are at the level of individual strains. So here shown for some of the top phylotypes in the Sphingomonidaceae, Sphingomonias and Pseudomonas are the two most common bacteria that you find in Arabidopsis leaves. So here is one phylotype that is much more common in northern latitude. Here's another phylotype that has the opposite pattern. This shows here on a map the behavior of this phylotype from the Sphingomonidaceae, which again is much more common in the north than in the south. So in the north, it makes up 20% of all the microbes in leaves. We then ask what explains the difference of these different clusters, and looking at different environmental factors, we found that PDSI, Palmer Drought Severity Index, was the best predictor of plants having either cluster 1 or cluster 2, and this is just shows on a map how Palmer Drought Severity Index is distributed across the map. So higher chance of drought in the north than in the south. That will not be surprising. And this really jived very nicely with our earlier work where we had shown that drought selects on host genetics, so both terminal drought and then response to drought, to milder drought across the range. So there seems to be selection of drought on both microbiome and the host genetics. And some of this is likely selection directly on the bacteria or on the microbiome, but a significant fraction is apparently mediated by genetics. And so when we ask what are the immune genes that differentiate the northern and southern cluster, voila, our top hit turned out to be ACD6. So we did not go in there expecting this this at all. But so top hint again is ACD6. So this really seems to hint that yes, ACD6 is important in the wild. Now, this is of course correlative and rather indirect evidence. Can we show directly that ACD6 is important in outdoors? So Derek Lundberg, who now has his own group in Uppsala, very simple experiment. He grew plants with contrasting ACD6 alleles, either in the greenhouse or in the field, in either Tübingen or in Zurich. And in Zurich, they are protective sites where we could actually grow genome-edited plants. And so this is results from RNA-seq, and you see nice differentiation between the field and the greenhouse. Well, you also notice that in the greenhouse, there's a nice separation between genotypes that have contrasting ACD6 alleles. However, the same material, when we look at it in the field, all these differences disappear, and both in these HIF lines and then with these genome-edited lines. That was, of course, really disappointing that we lost all phenotypic difference. So phenotypic difference are clear in the greenhouse, phenotypic differences disappear in the field, molecular differences disappear in the field, also content in hormones, which Derek profile disappears. We thought, well, maybe we sampled plants that were already adult and mature. Maybe ACD6 matters at least early on, so that there was a memory, if you will, in terms of the microbiome, since we had tied ACD6 to microbiome in a plant sample from the wild. Unfortunately, this was also really disappointing. We have a nice separation between plants that are grown outside and plants that are grown in the greenhouse. But if you look here at genotypic contrast, you see that they are all over the place, that there's no separation by genotype. Also, perhaps of interest, when we look at the greenhouse plant, at the field-grown plants, they actually cluster together with wild plants of unknown genotype, but that were sampled directly from the wild around Tübingen. So something happens here where the microbiome is different in the field and the greenhouse, but ACD6 is not what makes the difference here. And finally, then Derek also looked at potential fitness consequences. And as expected, in the greenhouse, the Columbia allele makes many more seeds, but in the field, these differences in seed production go away. So where does that leave us at? Well, it's a stark reminder that it's really difficult to find effects of single genes when you look across a broad range of samples, and that the effects of single genes are often probably limited to very specific environments and very specific situations. And this is also clear from what has been published here. Is this paper that I really highly recommend you read, from colleagues who used to work for Corteva or still work for Corteva, and describes the biotech program of Corteva, where they had tested over 3,000 DNA cassette constructs in something like 20 years, and essentially all of them failed. So none of these single genes made a difference in the field. How can we explain this failure? Well, on the one hand, I think we have become really good at explaining what goes on in individual processes. So we know a lot about the genes, proteins, and where they're expressed and all that, and their interactions for all these individual processes. And that's what we typically can isolate in the lab. But then when we move to the field, all these processes operate at different levels. They are very sensitive to the environment. And I think where we fail at is trying to understand how the signal is integrated between these different processes to then result in yield, which is typically the trait that we in both agriculture and in evolutionary biology. So what I want to leave you with is really this main conclusion here, that laboratory genetics can be simple. It can result in these really beautiful stories, but then real life ecology is anything but simple. And therefore, my favorite quote in ecology comes from this book by Rosemary and Peter Grant, How and Why Species Multiply. It's the last sentence in the book, that nothing in evolution makes sense except in the light of ecology. And I'll leave you with some nice pictures here of what it looks like when you look for Arabidopsis in the wild, and I'll be happy to take questions.